This video, we're going to take a look at Chapter 18, Objective 8. Objective 8, we're going to look at the different blood disorders that are going to be on your test. There's a lot more than what we're going to go over. So the first one we're going to talk about is anemia. Now, there's many different kinds of anemia, but for test purposes, we're going to go ahead and go with the standard, which is the number of red blood cells is deficient, which means your blood does not have enough red blood cells. That could be caused from many different things: blood loss, such as ulcers, hemorrhoids, excessive bleeding during menstruation and childbirth, caused by faulty or decreased red blood cell production, such as sickle cell anemia, iron deficient anemia, vitamin deficiencies, can all lead to red blood cells that aren't formed correctly or just a decreased amount of them. Or it could be from excessive destruction of red blood cells. So any of these could lead to red blood cells being deficient, and that's called anemia. Now, with sickle cell anemia, it is a genetic disorder caused by the production of abnormal hemoglobin molecules. So that's the big takeaway there that you need to know. And get a color you can see. So with sickle cells, you can see that they are not normally shaped. Remember, red blood cells are supposed to be biconcave discs. This normal hemoglobin right there, the normal red blood cell, but because the hemoglobin is abnormally shaped, it's going to cause the red blood cell to be oddly shaped, and it's going to have a sickle shape. And as you can see, it is because of its shape, it can clog up blood vessels and prevent blood flow downstream. Now, the genetic part, it is a genetic disorder. So what you're looking at here, you've got the blue side, which is going to be the healthy gene, and then the orange is going to be the recessive damaged gene. Now, in the father and mother, both with sickle cell trait, they're carriers of the disease. They don't actually have it. But when they have offspring, you can see that a fourth of the time, 20% of the time, if this baby gets the good gene from dad and gets the good gene from mom, they can be a healthy child. If you have a situation here where one good gene goes to the child and then the bad gene goes to the offspring, you could end up with, again, carriers of the disease. Now, in a child with full-blown sickle cell anemia, they're getting both bad genes, one from mom, one from dad. And so this would be a person who manifests the sickle cell disease. Just showing you kind of what normal hemoglobin looks like. Believe it or not, this is normal. This is the line that you're going to read. So the DNA should be GAG on one side. The other side, the other strand of DNA would read CTC. Now, if you notice with the sickle cell, this T right here should be an A. So it's going to cause one mutation. That's all it takes to cause sickle cell. And that order of nucleotide sequence is going to lead to a specific amino acid. So GAG is going to produce the GLU amino acid, and then GUG will produce the valine amino acid. So again, which would cause a genetic defect in the hemoglobin. You can see that the sickle cell hemoglobin molecule, because of that one amino acid, is going to change the shape of it. Iron deficient anemia. This is the most common. Its available iron is insufficient, so you don't have enough iron to allow production of the heme molecule. Remember, the iron is at the center of that heme molecule, and there's 4 heme molecules in every hemoglobin. So the problem with why you have not enough iron in the body is the person is unable to absorb and transport the iron. So for example, this is, I don't want to say it's a digestive issue, but you're going to get iron from the foods that you eat. And if you can't absorb that, that could lead to iron deficient anemia. And you can look up here in that actual slide there, and you can see that the center of those red blood cells looks more white than it should. So very, very much indicating that there's not enough hemoglobin due to, or iron deficient hemoglobin, not enough iron to make the hemoglobin molecule. Megaloblastic anemia, this is caused from a vitamin deficiency. Take a look at the size of the red blood cells. Megaloblastic, huge. So it's a deficiency of vitamin B12 and or folate. So those are two vitamins there, and you're deficient in one or both of those. Take a look at the normal size red blood cell and a red blood cell in a person who has megaloblastic anemia. This is a vitamin deficiency due to lack of eating the right foods that provide those vitamins. So it's not that you're eating the right foods and you're not getting the vitamins absorbed. It's that you're just not getting the foods correctly. And if you don't have the right foods, then you're not going to be able to get the vitamins. So it's a dietary issue. Pernicious anemia. Now, this is also a vitamin deficient type of situation, but in this one, it's not that you're not eating correctly. It's that the vitamins that you are getting in through your diet aren't being able to be absorbed. So you still have a lack of vitamin B12 in this one, but you're getting the vitamin B12, but it can't be absorbed through the small intestine into the bloodstream. This would be potentially with patients with Crohn's, where they've had problems with their small intestine, or they've had part of their small intestine even removed. Intestinal parasites could be a problem. AIDS could be a problem. So these are all potential diseases that can lead to vitamin deficient anemia for pernicious anemia. Aplastic anemia. So with aplastic anemia, you have a deficient number of red blood cells, stem cells. So those hematopoietic stem cells, you just don't have enough of those. So if you don't have enough stem cells, then you're not going to be able to get any. If you notice the big X's here, you're not going to be able to get any of the blood cells that you need. Not only just the platelets, the red blood cells, but also the white blood cells. This is often inherited, could be triggered by mutation from radiation, medication, chemotherapy, or an infection. Aplastic anemia. Thalassemia. This is another issue where we have a misshapen red blood cell due to a misshapen hemoglobin molecule. Now, this is different than sickle cell anemia, but it's still going to end up being a malformed red blood cell. So these are not sickle shaped. They kind of look like squares. You can kind of see them here. They're just not the right shape at all. And that's thalassemia. And there's different grades of thalassemia. So it just depends. You can have, without a mutation, you've got enough hemoglobin, so that's no thalassemia at all. Then you've got with one gene inherited, you can see there's a problem there. And then with two, but both genes inherited, one from mom, one from dad, you can see that this is also a person who's going to present with thalassemia. So again, the progression of the maturation does not happen correctly with red blood cells. Most severe anemia is called Cooley's anemia. So if it's Cooley's anemia, that's a person that's got the severe form of thalassemia. And then it's an inherited condition, mainly from these people here. Wow, that's a lot of red blood cells. So this is polycythemia. We saw what that looked like on a hematocrit and saw that, there's a lot more red blood cells than there was plasma. You can see under a microscope slide, there's a lot of red blood cells here. So this is polycythemia. Can occur in dehydrated individuals due to decreased plasma volume, not enough water, for example, or it could be due to an excessive production of immature erythrocytes, red blood cells. Leukopenia. Leuco means white, penia is going to mean small. So this is a small number of white blood cells. And if you look at the smear here, this field of view, you don't see any white blood cells. I see a platelet, but I see a lot of red blood cells. I don't see one white blood cell in that field. So that's very abnormal. Leukopenia, not enough white blood cells. Leukocytosis. Leukocytosis is too many white blood cells. And you can see in both of these smears here, there's a lot of white blood cells in that field. The problem is, and you would think, oh, well, that's a good thing. It's good for your immune system to have a bunch of white blood cells. You're going to fend off infection really well. Well, the problem is these white blood cells are non-functional, so they're just taking up space. Two major types of excessive proliferation of the white blood cell is called leukemia. You've probably heard of that. That's cancer involving an abundance of leukocytes. And then lymphoma, this is masses of malignant T or B cells, white blood cells, specifically T and B, that collect in the lymph nodes, spleen, and liver. So leukocytosis. Thrombocytosis, too many platelets. Remember, thrombocytes are platelets. So thrombocytosis is too many platelets. And you can look in this smear here, all those little bitty purple dots, those are platelets. And you can see there's a lot more in this smear than what there should be. It's caused from unwanted blood clots, thrombosis. That's what the blood clots are called. Thrombocytopenia, again, penia is going to be too small. So in this one, we don't have enough platelets. So thrombocytopenia is going to be too few platelets, which causes a problem because the person's blood is not going to be able to clot, which means the person can literally bleed to death quicker. Hemophilia. Hemophilia is not producing one or more of the clotting factors. It is a genetic disorder, so this is inherited. You can see right here, hemophilia A is due to a lack of clotting factor 8. Again, that's just one of them, okay, not all of them. There's many different parts of those clotting factors. And again, missing one can result in a condition known as hemophilia. Thrombosis. Thrombosis is excessive platelets, which are going to then increase the risk of clot formation. A blood clot is called a thrombus. Once that blood clot is now mobile and is traveling through the bloodstream, it is called an embolus once it starts moving. So the thrombus is an aggregation of platelets. Again, it's going to be sticky, so it's going to collect a lot of red blood cells and white blood cells all within this big, huge mass, which, as you can see, as this blood clot keeps growing, it could close off that entire vein. That is a vein right there that we're looking at, and can prevent blood flow downstream from that clot. Now, the embolus, like I said a second ago, is a portion of that clot that breaks off and starts floating through the bloodstream. And that's bad because as it goes into narrower and narrower blood vessels, it can then prevent lack of blood flow to certain areas. This embolus could get caught in the lung, prevent ability to breathe and get in air. It could go to the brain, cause a stroke. It could go to the heart, cause a heart attack. So again, that traveling embolus is not a good thing either. And that concludes Objective 8, Chapter 18.